The overall goal of this procedure is to generate telencephalic neurons from human embryonic stem cells, or hESCs, by going through checkpoints which are similar to those observed during human development. This is accomplished by first allowing hESCs to spontaneously differentiate in suspension in hESC medium, and then transferring them into neural induction medium to differentiate more selectively. Next, embryoid bodies, or PSC aggregates, are allowed to attach onto tissue culture treated plates, where the cells expand and differentiate into a monolayer with the centers forming neuroepithelial cells morphologically similar to columnar cells. Next, the neuroepithelial cells are isolated and placed in suspension cultures where they form neurospheres. Finally, the neurospheres are plated onto coverslips where they can further differentiate. Ultimately, results can be obtained that show the specific types of neurons formed through immunostaining or electrophysiology.
The implications of this technique extend towards a wide variety of genetically inherited diseases, because patient-specific neurons can also be generated from induced pluripotent stem cells, where the mechanism of action as well as potential therapies can be elucidated.
Though this method can provide insight into the development of telencephalic neuronal subtypes from pluripotent stem cells, it can also be applied to other systems, such as human diseases related to telencephalic neurons.
To generate human pluripotent stem cell aggregates, begin by culturing human pluripotent stem cells on mouse embryonic fibroblast feeders in hESC medium supplemented with basic FGF for 5 to 7 days. The colonies will be big, but still undifferentiated.